In our final section on the endocannabinoid system, we will discuss the role of the ECS and its main functions within the human body. The role of the endocannabinoid system is to maintain homeostasis within the body. Every major system within the body is controlled and influenced to some extent by the actions of the ECS. The five main words used to describe the actions of the ECS are eat, sleep, relax, protect, and forget. These are the five main roles the ECS plays in maintaining homeostasis within the body. Eat refers to appetite regulation. Sleep to insomnia management and the control of REM and NREM sleep. Relax refers to both physical and mental relaxation. Protect refers to the ability of the ECS to protect neurons from excitotoxicity and other insults. Forget refers to the role the ECS plays in proper memory storage and extinction. When you look at the five main functions the ECS serves, it is no wonder that the discovery of this system has led to such a surge of medical research and interest in the system and its role in the body. We are only just starting to uncover the nuanced way in which the ECS plays its role in homeostasis. The role of the endocannabinoid system in appetite regulation is well understood. External stimulation of the ECS is known to have a positive effect on appetite. In preclinical studies, it has been shown that endocannabinoid levels are increased in the hypothalamus during fasting and return to normal levels after food consumption. In fact, the role of the ECS in appetite regulation formed the basis for synthetic THC being developed as the prescription drug Marinol, used to treat nausea, vomiting, and wasting syndromes in cancer and HIV patients. There has also been significant interest in utilizing the role the ECS plays in appetite regulation in the development of anti-obesity medication. The prescription drug rimonabant, a synthetic CB1 receptor antagonist, displayed remarkable anti-obesity effects. However, it had significant psychiatric side effects, which led to its withdrawal from the market, further elucidating just how little we truly understand the nuanced effect of ECS manipulation using external agents. The endocannabinoid system has been closely linked with the sleep-wake cycle. Studies have shown that blocking the CB1 receptor promotes wakefulness, whereas the administration of endogenous anandamide enhances sleep. It has also been noticed in studies that endogenous cannabinoid levels begin to rise halfway through the sleep cycle, presumably to both facilitate non-REM sleep and in preparation for regulating morning appetite. Interestingly, in sleep deprivation studies, endocannabinoid levels were well above normal in sleep-deprived patients, indicating the body's attempt to regulate a dysfunctional sleep cycle through the ECS. There is still much to be learned about the endocannabinoid system when it comes to mood regulation, but we do have a good understanding of some basic concepts of how the ECS controls mood within the body. In studies, augmentation of endocannabinoid signaling has shown to decrease anxiety, whereas blocking or deleting CB1 receptors significantly increases anxiety levels, as seen with rimonabant, the CB1 receptor antagonist previously discussed. We also know that dysregulation of the ECS has been closely tied to mood and psychiatric disorders. The role of the ECS in mood regulation remains an area of great interest, and further studies will be needed to elucidate the nuanced way the ECS regulates mood, anxiety, and its potential role in the treatment of psychiatric disorders. The role of the ECS in regulating pain is one of the most well-understood roles of the ECS within the body. Clinical evidence points to the involvement of multiple receptors when it comes to the ECS regulation of pain, including not only the CB1 and CB2 receptors, but also the TRPV1 vanilloid receptors as well. Administration of cannabinoid receptor ligands has been shown extensively to produce analgesia in both acute and chronic models of pain. The ability of the ECS to mitigate pain through both central and peripheral mechanisms makes it a very attractive target for patients using cannabinoid medications to manipulate the ECS for pain relief. One of the most prolific roles of the endocannabinoid system in the body is its effect on the immune system. The ECS can both suppress and activate immune cells depending on where and how it is activated within the body. The ECS has been shown to induce apoptosis in immune cells and downregulate both innate and adaptive immune response. Studies have also suggested that the administration of endocannabinoids or the use of enzyme inhibitors that increase endocannabinoid levels leads to immunosuppression and a decrease in immune-mediated organ damage, which has led to significant interest in the manipulation of the ECS for the treatment of autoimmune disease, inflammatory disorders, and hepatic fibrosis. The role of the ECS in memory is very important for the day-to-day function of humans. If we were to remember every single thing that happens to us on a day-to-day basis, the volume of information would be completely overwhelming to our brains. By the same token, the ability to forget harmful or traumatic memories also helps protect our brains from overstimulation or the formation of traumatic disorders such as PTSD. One of the most fascinating functions of the ECS when it comes to memory is the ability of endocannabinoids to work specifically and locally inside the brain. It is believed that a few cells might be under the control of physiological activation of the ECS, whereas neighboring cells might remain outside the influence of endocannabinoids. The time of activation of the ECS can be very short, thanks to very efficient systems for the degradation of endocannabinoids on demand. It is therefore possible that during specific neuronal activity, such as the processing of memory, the ECS is activated in a limited number of brain regions for a very short time frame in order to exert its effects. This helps explain why the generalized and long-lasting action of exogenally administered cannabinoid agonists do not work the same way within the brain as the physiological ECS and can even exert opposite or deleterious effects. As with other neurotransmitters, dysregulation of endocannabinoids and the endocannabinoid system can also result in significant pathology. Clinical endocannabinoid deficiency disorder was first hypothesized in 2001 by Ethan Russo and is based on the concept that many brain disorders are associated with neurotransmitter deficiency. Russo hypothesized that a similar pathology may occur with endocannabinoid deficiency. Knowing that endocannabinoids play a significant role in pain, digestion, mood, and sleep, the role of a deficiency in patients with migraine, fibromyalgia, and irritable bowel syndrome was theorized. These specific syndromes were chosen because none of them have a definitive diagnosis other than one of exclusion. They all manifest hyperalgia, and patients with these syndromes display an elevated evidence of anxiety and depression, with a significant level of comorbidity between the different syndromes. Russo also theorized that patients with these syndromes had an endogenously low level of circulating anandamide and 2-AG within the body, which was later verified with clinical lab studies and shown to be an accurate assessment and hypothesis of these disease states. It is a logical conclusion that there are more syndromes humans experience that are closely tied to dysregulation of the endocannabinoid system. ECS dysregulation has also been associated with cannabinoid receptors. Altered expression of CB1 receptors has been observed in various neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and Huntington's disease. The activation of the CB1 receptor has been shown to prevent the formulation of amyloid beta induced neurotoxicity in several cell models. In addition, the activation of CB1 receptors has been reported to be beneficial in animal models of Alzheimer's disease associated with memory deficits and cognitive disorders. In 1993, decreased expression of the CB1 receptor was first reported in the substantia nigra of Huntington's disease patients. Further studies revealed a progressive loss of CB1 receptors as an early sign of Huntington's disease, which occurred before the onset of actual neurodegeneration and hastened the worsening of Huntington's disease development. CB2 receptor dysregulation has also been investigated for playing a role in multiple disease pathologies, such as traumatic brain injury, Alzheimer's disease, autoimmune disease, multiple sclerosis, polycystic ovarian syndrome, osteoporosis, endometriosis, asthma, and more. The better we understand how the dysregulation of CB1 and CB2 receptors affect the disease states within the body, the more we can look at the pharmacological modulation of receptors as a novel, therapeutically exploitable strategy. To delve even further into dysregulation of the endocannabinoid system, scientists have begun looking at single nucleotide polymorphism or genetic mutations within the ECS and the disease associations they have within the body. This chart represents a small summary of the different studies being done into how these genetic polymorphisms are associated with disease and represent a novel therapeutic strategy for many common syndromes. In summary, the endocannabinoid system is the largest receptor system in the human body, and its role is to maintain homeostasis of the body's systems. The ECS is comprised of CB1 and CB2 receptors, the neurotransmitters anandamide and 2-AG, the enzymes that form them, and the enzymes that break them down. Signaling within the ECS acts in a retrograde manner, inhibiting the signaling of the presynaptic neuron, which has widespread clinical applications. Dysregulation of the ECS has been associated with many disease states and presents a novel therapeutic opportunity for exploration. This concludes the endocannabinoid system learning module. Thank you for joining us.